Alrighty folks, I'm back yet again. We're going to do quite a short video this time. I don't want to... Last one I did was 2 hours long, and that was, as you can imagine, a bit of an absolute pain. So why don't we just do something, a bit more down to earth? And I've decided I'm just going to do the first paper of high human biology. So this is the multi-choice. So it should be nice and simple. Um, but yeah, even though this is quite easy to mark because it's just literally choose a letter, I thought it might be helpful for folks to like get an idea of why each of these the options are correct. Well, we'll just see how it goes, eh? So, question number one. Which row in the table matches the type of cell division that occurs in germline cells? And maybe if you bring a highlight, I'm not a highlighter person, but you might want to highlight it. Keywords like germline, with a description of the gametes produced. So we've got these options here. Now, germline cells, important to note that they can actually do both mitosis and meiosis, okay? Because when they do meiosis, right, they turn into haploid gametes. So I'll just go and write that out. Maybe it'd be better to use text tool, eh? Lloyd, but also they can um, use mitosis to divide as well, but that will create diploid cells, and that's because they'll just be creating other copies of themselves. Okay, so what can we go from here? Well, go on here. We can start ruling stuff out that we just don't think is correct, eh? Type of division, mitosis, gametes produce diploid. That's a gamete. You are not germline cells are not gametes, so we can rule that. It would be true if it was like cell produced. That could be true, but it's a gamete. Gametes are always haploid, so actually we can ditch these C and A. So because they're diploid, we want haploid. But look at this, right? Mitosis. We already figured out mitosis produces diploid cells. So it can't be that. So it must be D. Oh dear. Okay. Which of the following statements about DNA replication is correct? Fragments are formed on the blank, and it will go to all these different answers. So this is talking, there's two types of DNA replication. Well, not really, but yeah, there's two different ways DNA can be replicated. It depends on the strand. So on the leading strand, I'll put that as LE on here, it's replicated continuously because the free end is exposed. But on the lagging strand, I'll put that as LA, it has to be replicated in fragments because it can, remember, DNA polymerase can only add DNA nucleotides to the free end. So it's perfectly fine and down the leading strands, but on the lagging strand, it has to make loads of little wee fragments, alright? So that means we know can't be leading strands because that has fragments only form on the lagging strand. So A and D are out completely. And then it says, the only other thing it says is joined by ligase or joined by DNA polymerase. And yes, this is a bit where you could, I do see how people get this wrong, because DNA polymerase still adds nucleotides, but it doesn't join the fragments together. It simply places them, and then the glue, if you will, is ligase. Okay, so our answer for that is B. Next up, PCR, which is polymer chain reaction, was used to amplify, which means just duplicate. Yeah, whenever I put amplify, I don't know why, because I think it's just a bit of wordy, but amplify means just duplicate a region of DNA. Of the 5 cycles, 32 copies were present. Calculate the number of additional copies, and it's important because they put it in bold. Present after a further 4 cycles. So remember, this is quite a mathsy one actually. They additional copies after 4 present cycles. So if you're maths like me, you'll notice that, hey, that's green, we don't want it in green, do we? 2 to the power of 5, because remember, it doubles each time, that equals 32. That's cool. So Over 4 further cycles, that's literally just time going up to. That's not a 2, that's a 3, ain't it? That's going up to 2 to the 9. So you can just work that out. And I'll go and pull out my calculator on my phone. But that's not you. Just to quickly work this out. But 2 to the power of 9 is 512. So yeah, if you're not mathsy like me, that's fine. But you can just think about, like, you just times 32 4 times. So this also means you can just go 32 times 2 times 2 times 2 times 2. Okay, that'll give you the same answer. So 5 and 12 must be D then. Oh ho ho, wrong. Because remember, additional copies, which means we do not include the original 32. So we actually go, oh man, this is getting a bit of a mess, isn't it? We're actually going to take away 32 from 512. And what does that get us? Yeah, that's right. 480, if you can read that. So it's going to be C. So yeah, no, this is a bit of a trick question. So don't worry if you don't get this right the correct first time. Like, I think this has caught me out as well. Anyway, we'll onwards and upwards. Question number 4. The list shows the nucleic acid molecules involved in gene expression within a cell. We've got RNA, 2 is mRNA, DNA, TNA. Which of these molecules are required for translation? So, translation, remember, is the process of you have an mRNA strand, pretend that's a black line, it's got its bases on it. It's the process of adding tRNA. I'll do that in a different color just to see, adding tRNA molecules on top of it with their linked amino acids, right? And the amino acids are put in yellow. Okay, so right off the bat, we know that we have to have tRNA and mRNA. But remember, where does translation take place? That's right, in the ribosome. Okay? And we need the ribosome for translation to happen. And rRNA, you can remember our ribosomal RNA is a part of the ribosome, so we need it to happen. But DNA, that's all the way back in transcription. We don't actually need DNA for this specific bit. We just need RNA, mRNA, and tRNA. So the answer would be C. Oh, there we go. Alrighty, we're moving on to 5. A student set up a test tube containing 5 centimeters cubed of milk and 1 centimeter cubed of the enzyme trypsin. The milk became clear, and the student concluded that the white milk protein had been broken down by the trypsin. To show that the trypsin caused the milk to become clear, control tubes should contain what? Well, this is an experimental question, but basically what you want to find an appropriate comparison. Okay, so I guess we can just do this process of elimination. 5 centimeter milk, and that's looking pretty good. I'm not gonna say definitely, but that looks decent for me right now because it's the same volume as the original thing, but it doesn't have the active enzyme because that's what we wanna show. 5 centimeters is still water in the trypsin. That's not what we want to show. We want to show that the protein be broken out in trypsin. That's not going to show us anything. It's it's just water. Boiled and cool milk and trypsin. I don't know what they're on about there. Boiled and cool milk, that should be break down enzymes, but that won't show that the protein's been broken down by this specific enzyme. And yes, so yeah, we've boiled it, but we've also got no enzyme. So yeah, all these are quite wacky. But A gives a proper through line because if we compare these two, it's the only difference is whether we have the enzyme trypsin in it, and that will show if it breaks down or not. Okay, hope I'm not going too fast for you folks. 6. Part of a metabolic pathway used to produce the amino acid isoleucine is shown. It's got this formula here. Isoleucine is a feedback inhibitor for the enzyme threonine denatase. Okay, so basically that what means is that if we go on this diagram, this is going to bind to this enzyme here and cause it to slow down. the relative slowdown. The statements refer to substances in the metabolic pathway. Isoprene binds to thermodynamics. Well, given that what I've just said, that's probably true. Let's face it. Threonine dianimate lowers the activation energy required to convert threonine into carbohydrate. Well, remember, enzymes are biological catalysts, and especially if you do chemistry, catalysts lower the activation energy, but enzymes, and therefore enzymes, will lower the activation energy of reactions. That's how they speed them up, essentially, right? So that's got to be true as well. Ketobutane is a substrate of isoclinic. Well, isoclinic is not an enzyme, right? So how can anything be a substrate of it, right? So that's garbage. Because it's a product that gets turned in by another reaction. It's not the substrate, you know. So 1 and 2. So it looks like I think it's going to be B. Oh, it is B. Right, moving on to 7. The table shows the effects of a competitive inhibitor on an enzyme's activity. So we can see here, as you increase the concentration of the inhibitor, enzyme activity goes down. Whoa, crazy. Predict the concentration of the inhibitor when half the active sites in the enzyme are occupied by the inhibitor. Okay, so we're going to have to look at this. Enzyme activity when there's nothing. That means all the enzyme sites are binded to the substrate. But then when you see add up, that means 40% of all the enzymes are going to be binded to something else. And then 60%, 80%, and 100%. 100% are binded to this inhibitor. So we want half. Now half, that means 50%. Which I'm sure you all know. I don't want to dumb it down too much, but we'll just make it clear on this. And so we want to look for enzyme activity that's at half. But oh my god, it doesn't say. It must be in between this, which means must be in between here, between 0.10 and 0.20, or 0.1 to 0.2. Okay, and if we go down here, every single one is higher and is not in the range, apart from A. So 7 is A. That's a beautiful wee circle there, ain't it? Okay, we're moving on to 8. An investigation was carried out into the effect of substrate concentration on the rate of reaction in the presence of a competitive inhibitor. Whoa, what a mouthful. Which of the following would ensure the results were valid? Oop, I'll go and highlight that just because validity means the experiment actually makes sense and it will produce a good result rather than just being insane, you know. What are these? Repeating the investigation using a non-competitive inhibitor. B is keeping the inhibitor concentration constant. C is repeating the investigation with each concentration of the substrate, and D is keeping the substrate concentration constant. Okay. So I just get myself a wee stretch. So whenever someone's repeating and validity, that's not, those two don't usually go together because repeating is more about reliability, showing that you can repeat the experiment and it's going to do the same thing over and over, which is reliability, it's not validity. So how about we get rid of those two to start out with? So we're left with B and D. So it's either keeping the inhibitor concentration constant or D keeping the substrate concentration constant. And it's the effect of substrate concentration on the rate of reaction in presence of a competitive inhibitor, which means keeping the substrate concentration is constant doesn't make any sense because we're changing it in the experiment. You know, it's like you're doing an experiment to see how temperature affects the rate of reaction, but then you keep the temperature the same, so that's just going to give you a garbage experiment. So D can't be right. So it has to be B. Keeping the inhibitor concentration constant because you're changing one thing, but in science you can't change. You really shouldn't be changing more than one variable. So keep your inhibitor concentration constant. Number 9. During aerobic respiration in a cell, 1 molecule of glucose yielded 38 molecules of ATP. During respiration without oxygen in the same cell, 1 molecule of glucose yielded 2 molecules of ATP. Calculate the percentage decrease in ATP yield per glucose molecule when this cell carries out respiration without oxygen compared to what it respires aerobically. Okay, well, this is a bunch of nonsense words for basically just a maths issue, right? So percentage decrease. So we want to find the change in these two. Well, that's easy enough, ain't it? So we can just go 38, take away 2 equals 36. And then we just do a percentage calculation. There we go, because it's like out of, like, if it was 100% decrease, then it would be nothing. So these two would be the same, but it's not. So it's almost the same, but it'll be it used to be quite large. And again, you're going to have to pull out your old dandy calculator, but 36 divided by 38 times by 100. It's not exactly, it's actually going to be 94.7%. Oh dear, that doesn't look like it's any of these, but it does round up to 95 because the SQA does like pulling that one on us, doesn't it? So there we go, and that means it must be C. I mean, you could also do some eliminations from other ones, like D. Like, you can't decrease ATP, that'll be a negative number. So just have a look at the results and make sure that they're somewhat logical. Okay, we're moving on to 10. The diagram represents some of the processes that occur in mitochondrion. Oh, and this is the electron transport chain, so that's going to be a good laugh, wouldn't it? Which letter represents ATP synthase? So this is the molecule that hydrogen ions go back through to generate ATP back through the inner membrane. Well, we can have a look at all these different letters one by one and see what they are. So I'm gonna go here. It goes back. It's the direction is this way, right? That's the ATP synthase. So yeah, D's looking pretty likely so far. But let's give the other letters better for that. But because it generates energy by electrons moving along there, so that means C must be electron, not ATP synthase. And these proteins are all going the wrong direction. Okay, well it looks like has to be D because it's the hydrogen ions going back through. I know it's a bit boring. I don't know if I did a good explanation. Go and comment if you got any questions. Which of the following applies to the skeletal muscle fibers that are most useful during a cross-country ski race? Cross-country, think about it. That is long term. It's a long time, right? So we want muscles that can do exercise for quite a large time, which means they probably have to be slow twitch, right? Because slow twitch are for like marathon runners, but fast twitch are for sprinters, right? Now, and then we just need to look at the properties of slow twitch and have a look at this. So slow twitch have lots of mitochondria because it uses a lot of aerobic respiration. So let's tick aerobic respiration. That's what we're going to use. Glycolysis are used in fast twitch because it's fast and easy, but you run out, right? We also have a large blood supply for slow twitch, and we have lots of mitochondria. And as you can see, there's only one box that ticks all of these boxes, and that's D. Because slow twitch have lots of mitochondria, a large blood supply, and primarily use aerobic respiration, not glycolysis. Okay, move on to 12. Which hormone found in contraceptive pills causing thickening of the cervical mucus? Alrighty. So this is one of those things that you're just gonna have to know, but it's, I guess, how you can go about it is you can say, oh well, it's thickening. So that must mean it's in the other half of the menstrual cycle after ovulation is finished. So it's probably not estrogen or follicle stimulating hormone because those kind of happen before then. It's not luteinizing hormone either because the surgeon LH actually causes ovulation, which is not what you want when you do contraceptive. So it's probably progesterone, ain't it? And it is one of the things you probably just need to remember, but progesterone, when it's produced by the corpus luteum, it causes thickening of the cervical mucus, just because that once it starts producing there, they don't want any more sperm getting into it. the reproductive tract. Ah, sorry. Right, we're on to 13. Which letter in the diagram indicates the site of testosterone production? Right, well, this should be fairly easy enough. I'm going to try and name all the organs, but C, I believe, is the bladder. And, well, we all know what the bladder does. It's no way it's the site of testosterone production, ain't it? B, I believe, is the seminal vessels. Correct me if I'm wrong, but it doesn't really matter because they don't they have nothing to do. And then A is the prostate gland. That's not how you spell prostate, is it? Ah, oh, peaks to this. I'll just use text. Right. Well, we already know that seminal vessels, prostate gland, bladder, all of them do not produce testosterone. But you know what does? Yeah, it's the testicles. Or the testes, I assume. Because that's where the interstitial cells are found. So it has to be D. And we're back. We're going for question 14. Cystic fibrosis is caused by a recessive allele. Yeah, make sure to remember that. That'll come up. That's quite important, actually. The diagram shows the inheritance of cystic fibrosis in 3 generations of a family. Okay. Which individual confirms that cystic fibrosis is not, keyword there, not, it's in bold, I don't know if I need to highlight it then, a sex-linked condition. So we're looking for reasons why it can't be. Well, I would suggest let's go through all these people and see if they tell us anything. Well, we can get rid of D. They are not affected by cystic fibrosis, so they can't tell us anything. What about C? Well, C, we can't tell anything either because even though it doesn't, it could be either way. Because remember, cystic fibrosis is a recessive allele, which means that the mother could be carrying a faulty X chromosome, right? I'll represent that as X0, and that's what caused it. Because he will only have XY, so he won't have any another X chromosome to override the faulty one. So it could be sex link in that case. So C doesn't tell us that it's not. And what about B? So it's worth noting for the mother of A, B, and of 1, they will have to have 2 copies of the thing because females have 2 X chromosomes, and it's a recessive allele, so they have to be homozygous for the cystic fibrosis. Okay, and then A is B is a male, so you'll have XY, and then A is a female, so they'll have XX. So B, it still could be say sex linked because he'll receive his Y chromosome from his father. So if they have a mother that's got 2 faulty X1, he's guaranteed to have it because the only X chromosome comes from his mother. But if A is affected but has 2 X chromosomes, but here the father's fine. His only X chromosome has got no mutations on it. And whoops, sorry, I just zoomed out of the way, confusing everyone. Which means that if this was sex linked, then A would have 1 good chromosome, 1 bad one, and since it's recessive, it should have overridden it. Which means A proves that cystic fibrosis is not a sex-linked condition. Ain't that fantastic? Moving on to question 15. Make sure to read this carefully, because I definitely didn't. The following steps are involved in the process of intrafertilization. In vitro just means fertilization on a petri dish, essentially, because in vitro means on glass in Latin. Yeah, there you go. There's your brain booster for today. So what I'm noticing down here is that we can eliminate half the answers by just deciding which step we think is first. I'm looking through here. Women is given drugs to stimulate ovulation. That should always happen. That should happen before eggs are surgically removed, because if they're not ovulated, then where are the eggs, right? So we know that 2 is the first step, right? And we can get rid A, and we can get rid of C, leaving us B and D. Just by doing that, you've got a 50-50 shot of getting the right answer. Okay, well, we can continue going through this. I think, well, actually, the only possible way is that 4 would have to be next, because those are the only two other answers. And then, again, 3 would be next. So, just check this makes sense. They're circularly moved, and then they're mixed with sperm and culture this. That makes perfect sense. Only difference between, and then, yeah, the same one that goes to 6, fertilization occurs. I mean, yeah, that's what happens when you mix sperm and eggs, ain't it? And the only difference is what comes next. Are they incubated first or are they implanted first? Well, notice on this, zygote and embryos. Embryo is a later step than zygote because zygote is the like the very first cell that happens after the fusion of the nuclei of the sperm and the ovum, and an embryo is a very small ball of cells. So that means 5 must come later. And yeah, it makes sense. You want to incubate something to get up to a decent size before you implant it, which means D is a bunch of nonsense and it has to be B. Ooh. Right, moving on to 16. Measurement of a child's cardiac cycle showed that systole lasts for 0.2 seconds while diastole lasts for 0.4. What was the heart rate of this child? Well, remember, cardiac cycle consists of 2 steps, systole and diastole, and that's the whole thing. So if we want to calculate the whole cardiac cycle, length of one cardiac cycle, we're going to add these two numbers together, right? So that means each cardiac cycle is 0.6 seconds, right? Okay, well, how are we supposed to turn this into beats per minute? Well, if each cardiac cycle is 0.6 seconds long, and then a minute is 60 seconds long, that means that if we divide 60 by 0.6, we can figure out how many beats happen in a minute. So, hopping onto your calculator now, I can't show this unfortunately, you'll get 100, which makes some sense, eh? 100 beats per minute. I'll try to do some lettering, eh? 100 beats per minute. And then, well, there's only one answer there. That's 100. So it's C. Moving on to 17. Peripheral vascular disease most commonly occurs in the arteries to the, well, it's peripheral, right? So no way is it going to be anything centralized. So we can get rid already heart, brain, and heck, lungs is pretty centralized as well. It's probably going to be leg. Yeah, so peripheral vascular disease, that's disease that affects arteries outside of the ones with lung and heart. You know, one of the things you just gotta remember, I'm afraid. Right, moving on to question 18. In a study, the percentage of people aged 55 years and older who use statins, now statins are drugs used to help with diabetes, okay, was recorded in 1990, 2000, and 2010. I'll just write diabetes here. Why not? Boom. Which of the following statements are correct? Between yada yada. Well, let's go and start eliminating some of these then. Process of elimination is your biggest friend here. So between 2000 and 2010, statin use increased in all age groups. Well, looking at this, that's provably false because for 55 to 64-year-olds, it did not change at all. So we can remove A from that from our list. Between 1990 and 2010, statin use in the 75 plus age group, the grey one, tripled. Okay, well, let's read the graph. So this is 2 above 10, and it's each box is 2. So that's 14. Oh my lord. There we go. That's 14 percent there. And then in 2010, what was it? It's 2 above 40, so that'll be 44. And it doesn't take a genius to work out that 3 times 14 does not equal 44. So I think we can eliminate that one as well. What about C? Between 1990 and 2000, statin use in the 65 to 74, so the white box, doubled. Okay, well, in 1990 it was 12. I'll do it in yellow just to make a slightly different color, eh? Okay, and then in 2000 it was 2 above 20, so that must be in 24. And I'll zoom in a bit just to probably turn my DPI down, but yeah, that's for... Well, and 12 times 2 does equal 24. So C is looking good, but just to double check, it's definitely C. We'll do D. Between 1990 and 2010, statin use in the 55 to 64-year-olds, so the black one, decreased. Well, as we've previously established, they've stayed the same. So that's got to be a dud. So it has to be C. Right, moving on to 19. Which row in the table matches the branch of the autonomic nervous system (ANS) with its effect? Okay, well, sympathetic. Remember, just write this out here. Sympathetic equals speed up, because it speeds up your heart rate and breathing rate, right? And then parasympathetic, remember, it's like a parachute. So it will slow you down, and I can't actually spell anything at all. No. Equals shoot, so slows down. Okay, so using that font of knowledge, we can have a look through this. So sympathetic increases testosterone, so that means more digestion. Well, if you're running away from a lion, do you really want to be... Is digesting that hamburger really your top priority? I don't think it's A for that reason. Okay, well, we're going on to B. Sympathetic decreases breathing rate. What? You want to be breathing faster when you're running away from a lion. That's useless. Parasympathetic decreases in execution. Well, when you're all done and dusted and you've finally caught that lion or something, you probably want to start digesting things again. You're all relaxed. Like, this looks like you're preparing for something, but no. So, for process of elimination, it must be D, but we'll just double check. Parasympathetic increases peristalsis. So peristalsis is fancy Latin gibberish for moving stuff around your digestive tract. So increasing digestion again. And yeah, when you're resting up, you want to be digesting your food, you know? So yeah, D makes sense. Number 20. Which of the following is not, keyword not there, a method of encoding information into long-term memory? Okay, well, encoding, remember, that just means like storing information into long-term memory. And if you've done in a studying of encoding information along memory, you actually recognize these 3 words, because those are the 3 techniques for improving encoding into long-term memory. Sorry, my mouth is really dry. You can probably hear it. So yeah, that means all these, they can't be true, because rehearsal, that's repeating information over and over again. Elaboration, that's adding extra detail onto information. Organization is organizing into meaningful groups. But all those help encode information long-term memory. So that's garbage. So it must be chunking. But why is it chunky? Because chunky does help with encoding. Ah, yes, but it helps with encoding information into short-term memory. So it has no effect on long-term memory. That's the wee trick they get you there. And I'll see if I can write short-term without looking like a complete idiot. Yeah, that looks about right. So that's why it's chunking. And chunking is like when you're reading a phone number, you don't go individual numbers. You put it in, instead of going 1, 2, 3, you go 1, 2, 3, 4, 5, 6. You know, you kind of combine them into wee groups to make them easier to remember. Moving on to 21. Split brain patients cannot transfer information between the right and left hemispheres of the cerebral cortex because the corpus callosum has been cut during surgery. In a study, a split brain patient was asked to press the forward against the barrier so that their left eye could only see to the left of the barrier and their right eye to the right of the barrier. The diagram shows the setup of the study. The patient was asked to look straight ahead, and then the 2 patient patterns were flashed briefly on the screen as shown. Which of the following diagrams represents where the patient patterns are interpreted in the visual areas of this patient's cortex? Okay, so I'm going to draw a diagram on this guy. Brains are really weirdly designed, right? So imagine these are your eyes, right? They actually go and connect themselves to opposite hemispheres. Who knows why they do this, but that's biology, mate. It makes absolutely no sense. So that means this pattern must be displaying on the right side, and this pattern, the straight line pattern, must be playing on the left side. So already we can get rid of that. B's complete garbage. And remember, each side only processes information from one eye. So these A and D of these sides processing both are junk because they just receive information from one eye. So it must be C. Ain't that correct? All right, we're moving on to 22, almost done. An individual displays a hypersensitive response to a foreign substance. Which row on the table identifies the cause of this response and the type of lymphocyte involved? So, hypersensitive response to foreign substance, that's fancy science talk for allergic reaction. One day I'll get good at handwriting. Okay, so it's like hay fever. People, I'm sure you know what an allergic reaction is. You're in high human biology. So that means we can get rid of autoimmune diseases. Okay, so we're already down to B and D. Now, how is an allergy caused? Well, it's when B cells they produce antibodies towards foreign subs that can be completely fine, but they have a sensitive response anyway. T cells they only basically mess around with killing like a virus and like performing apoptosis, which is killing of like what they perceive to be as damaged body cells. So they'll have no effect. They won't have many effects on foreign substances. They only deal with our cells. So it must be B. Okay, so 23, I've had to redo this because I really flipped up the first time, but don't you worry. During an epidemic, the R number refers to the number of cases of a disease that are directly caused by contact with one infected individual. During an epidemic, an individual becomes infected when the R number is 2. This means that during the first level of transmission, 2 further people will be infected, or each infected 2 more people during the second level of transmission. The total number of people infected of the 4 levels of transmission. So, we're going to make a nice diagram, mate. So, we have person number 1, patient 0, if you will. Okay? They're going to infect 2 other people. Those 2 other people, well, they're both going to meet someone, and they're both going to affect them. So they're going to affect 4 people. So that's one layer, that's the second layer. And you notice anything? Well, each time we're going to be timesing the number by 2. So 4 people, they infect 8, and 8 people, they infect 16. So I'll flash up a mathematical formula that does, if you want, you can just throw it into your calculator, but this is the general idea. If you add up all these, so 1 plus 2, that's 3, 3 plus 4, that's 7, 7 plus 8, that's 15, and 15 plus 16, you get 31. And wouldn't you believe it, that's our answer. Moving on to 24. The diagram shows a lymphocyte releasing proteins it has produced. Which one on the table identifies the type of lymphocyte and the proteins produced? Well, okay. So, I don't know about you, but I've never heard about T lymphocytes doing anything in producing. I mean, actually, the lymphocytes can produce cytokines, to be fair, but there's no cytokines on the thing, so we can eliminate those. So, do B cells produce antibodies or antigens? Well, antigens, I'll remember, are the like spiky bits on the south side of pathogens. They allow, or cells actually, they allow cells to recognize them. So I guess they technically do produce antigens, but definitely they don't release them out into the wild. They just have them on their surface. So D doesn't look right well. It must be antibodies. And yeah, I mean, I think this is quite easy. I mean, look at this. Like, those are clear as day antibodies. Antibodies are Y-shaped proteins, remember? So 24A. 25. The list contains statements about factors considered during the vaccination of a population: the type of disease, the population density, effectiveness of vaccine, the percentage of non-immune individuals. Which of the statements refer to factors that can affect the herd immunity threshold? Well, let's go through all these. Can the type of disease affect the herd immunity threshold? Absolutely. It's usually transmission of disease, because if the disease is super duper transmission, like COVID, goes everywhere, you'll need to vaccinate more people. But if it's not very transmissive, like trying to think of a few ones that people would actually know, but say it's not. I mean, Ebola wasn't ludicrously transmitted. There was outbreaks of it, but it wouldn't need as many people to be vaccinated because it could be contained easier. It doesn't matter. And same thing goes with population density. That will also affect the herd immunity threshold. Because if you have tons of people crammed in one room, for instance, diseases are going to spread like that like wildfire. You need to vaccinate pretty much everyone. But if you have like a person per square kilometer, then they're not going to infect people really rarely. So you can afford to have a lot less people vaccinated. And of course, the effect of the vaccine would affect how many people need to vaccinate. Because if they're only 10% effective or something rubbish like that, then you have to vaccinate loads more people, because then most of the people vaccinated, it's not going to work. But if it's 100% effective, you can get away vaccinating very little, because they will act as like barriers that they will not contract disease and won't pass it on at all. So that will affect. But what about 4? Percentage of non-immune individuals? Well, that won't actually affect the herd immunity threshold, because that's sort of what we want to change. We want to decrease the percentage of non-individuals so we can get herd immunity, right? The herd immunity is the percentage of people, like if this is the whole population, right? It's the percentage of people that you need to vaccinate up to so that everyone is protected, even if they're not vaccinated. So the percentage of non-individuals won't affect it really, because it's nothing to do with the thing. So yeah, it's going to be C, I'm afraid.